Come on. Si se puede, yes. So, there it is again. So, last time we talked about we were trying to get through 2.A.2, and the last thing we did with it is talk about electron carriers. One more time to review where we left off from there. The idea of oxidation and reduction. Let's say, for example, you look at the follow for photosynthesis and cell respiration. I'm going to write the photosynthesis one. Trying to make it tiny here. C6H12O6 plus 6 oxygens leads to 6 carbon dioxides and 6 waters. Now, when you look at what's happening there, electrons from glucose, right, are in between. We don't see that in the equation, but in between, electrons from glucose are being passed on to electron carriers. And ultimately, they are all dumped into water, which means water is, you can think of it as oxygen being reduced to water. So the electrons from this and the hydrogens will be dumped into oxygen, right? Meaning the oxygen gets reduced. Let me change the color of this so you guys can see the two reactions that are actually happening. So the oxygen is being reduced to water. Meanwhile, the glucose is being oxidized to carbon dioxide. You know, now we talked about how people have the misconception that that carbon dioxide becomes oxygen becomes carbon dioxide and vice versa during the process. But that's not what happens at all. Electrons and hydrogens carrying those electrons are dumped into oxygen. But in between, there's electron carriers that are going to carry electrons throughout the process. And we talked about some of those last time in photosynthesis and cell respiration. We have these guys. So this is an electron acceptor that shows up in cell respiration. That's an electron acceptor that shows up in cell respiration. That's an electron acceptor that shows up in both of them. And then we have the lead P, which shows up in photosynthesis. But the ultimate electron acceptor of cell respiration is also oxygen, right? Because oxygen gas is going to be accepting the electrons at the end of the ETC. So energy capturing processes are important because what life is trying to do is capture as much of the energy as possible rather than let it escape as heat. And so we have to have these efficient electron carriers. So where we left off is starting to talk about the light reaction of photosynthesis. So, and I was trying to start talking about the photosystems. My computer decided to be especially slow today, so bear with me. So in order to give you guys 50 minutes to work on the labs, we would have to 25 minutes before the hour? We would have to 35, yes. At 35 I have to stop. We'll do as much as we can till then. Cell energetics. Come on. Just do it!
Don't let your dreams be dreams.
Is he in jail right now?
Yeah, he is.
Why? No.
Is he really in jail?
Yeah, but why?
I don't know. He's always drinking too much.
Did you see the cannibal Shia LaBeouf?
Okay. Oh, this is awesome. Yes. The force awakens. So the actual last thing I talked about, I remember now, was the actual structure of the chlorophyll molecule. And I talked about how chlorophyll has the magnesium right in the middle, and it kind of like the whole molecule acts like a filter that focuses the light into the atom. and eventually causes an electron from the atom to absorb that sunlight and get bumped into a higher level of energy. So outside the atom altogether. Where is my PowerPoint that opened up? Nope, haven't opened up yet. But another thing I want to talk about, and it's going to be, I'll have a picture in the PowerPoint in a second, is that it's not just chlorophyll that does it. There are many pigments associated with photosynthesis, and what they do is that, think of it as that each pigment is trying to capture a little bit of that sunlight, you know? And so what will happen is that the sunlight will hit the first one, an electron will bump out of it, right? And then replace an electron that the second one lost. So it's like a chain. At the end of the chain, though, you have chlorophyll, right? So chlorophylls, when an electron bumps out of chlorophyll into the first electron carrier of the process, right? That becomes electron depleted, right? So an electron has to come to replace the electron that came through that one. And it could come directly from the water that's being split, or it can come from an electron that's bumping from another pigment. And then that pigment also becomes electron depleted, so another pigment needs to replace it, and so forth. So it's a chain. So each one of them gets hit by light, and these electrons jump to another one that needs it. Are you with me what I'm saying? But ultimately, they all need to get their electrons displaced. And that's water that's going to be doing that. When water splits up, two things are accomplished. And by the way, last class, we also talked about some of the structures associated with the process. Photosynthesis, though, occurs in two stages. Right? We have light reactions, or called light-dependent reactions, because they only happen in the presence of light. And those will happen in the thylakoid membranes. And then we have the light-independent reaction, also named the Calvin cycle, or carbon fixation, which rely on electron carriers, again, from so basically what the light-dependent reaction does is produce charged electron carriers. So we have the NADPH, right, which is what this gets once it remember I talked about last class. To really receive an electron, you can't just receive an electron, you have to form a new bond. And so that's why these things this one becomes ATP by bonding a new phosphate to it, and these all bond with hydrogen to become that FADH2, NADH, or NADPH. So you see that NADP get dumped into thylakoids, and the thylakoids recharge them to NADPH, which is then used up in the Calvin cycle to power it. So you can think of this as batteries that power this engine, right? So it's an engine powered by, it's a cycle or engine powered by batteries. But these batteries are recharged by solar power plant. So that's how I think of it. And ATP is necessary. So it's interesting that people think that photosynthesis produces ATP, but in fact, it's actually consuming ATP. All right, it's going to consume the ATP to put that energy inside of glucose. So it's the exact opposite of this. If we say that glucose gets oxidized to carbon dioxide, right, it's the other way around. So you're going to actually, carbon dioxide is going to become reduced to glucose. Right? So electrons from ATP and NADPH get dumped in the carbon cycle to charge up carbon dioxide to become glucose. You know? And as glucose gets formed, new bonds between oxygens and hydrogens and carbons are made, and that's why you also need hydrogens, which of course come from the NADPH. The H is carrying the hydrogens, right? And so that's why you go from carbon dioxide to something that's C6H12O6, because you are actually dumping hydrogens as well as you dump the electrons. All right, so these are some things. Another thing that happens is the opposite. Since water here in false aspiration was an electron acceptor at the end, all right? So since it's the opposite, water is the electron donator at the beginning of the entire process. Water is split in order to provide the electrons that are being dumped into the batteries. And you get oxygen out of that. Now, of course, that oxygen diffuses out of the thylakoids, out of the stroma, that's the liquid inside of here, and then out of the chloroplast, and then out of the cell, and then out of the leaf, right? And so the oxygen goes into the air. And of course, likewise, the carbon dioxide has to come from the air and in reverse. Now, so it's really two stages. So the first one, the EKs go over, and we already talked about these structures last time. We talked about basic structure of the plant cell in my videos that go over more detail. If you get lost, confused, it's too fast for you, make sure you watch the videos there and go into detail about it, right? Yeah, I even talked last class about each part of the leaf and what it's for. Does anyone remember what the Palisade Parenchyma does? It's mostly a heat sink, which means if you ever get to the lab, by the way, I try to set up hot water baths for those of you who want to do temperature modifications. But if you are interested in doing that, you should probably be watching your temperature to make sure it doesn't go too high. For example, this one's already too high. That one's already too high. Because I don't know, you guys said something about room temperature, and then 35, 45, 55 would be good. And there's two groups doing that, right? But this one already went up all the way to 60, so actually this one is at 80 already. I'm going to lower it a little bit. So I'm just saying, have one person of the group who's actually in charge, who's doing any of that, to sit right here and be monitoring that, because you guys have to play with the dials to make sure the temperature stays at the range where you want to stay. Because you want this to normalize at that temperature. So it's actually too hot right now. So I'm taking it out so that it can cool off. But we don't want it to become too heated. You have to put in a temperature setting and just let it sit there until it normalizes at that temperature. So that's the problem. So I put it at low 2 and 4. And I was hoping that it was going to equalize. But I don't know. Hopefully by the time I'm done with the lecture, it's sitting in the constant temperature. Let me see. No. That's why. Oh yeah, you saw that. Anyways, we also talked about surface area development ratio as far as leaves. If it was just as an absorption surface, leaves will have to be very large. But since they do gas exchange, leaves are also tiny. We talked about the stoma, the hole is in the bottom. We talked about some stomatic modifications. Some plants have no stoma. Why would you have no stoma? Because you're in the water. You don't need to do the diffusion through the leaves is how they get things. Why would you have stoma only on the top? When you're on top of water, so you need to exchange of gases through the upside. Why would you have stoma? A lot of stomas all over the plane, top and the bottom. You're not worrying about losing water. Why would you have stomas only on the bottom? You're trying to conserve water. And those will typically also have less stoma because they're. So give me a plant that you think will have less stoma. Yes? Cactus. Actually, cactus doesn't have stoma at all because they don't have leaves at all. Right? So that's another thing. Oh, no stoma, that means on the water. Well, or no leaves. Right? So yeah, don't just think. And we also talked about how they close that hole. Right? So when they get turgid, they close the hole. To become turgid, they got to gain water. To gain water, they have to pump salt inside, right? And then to open the hole, they have to pump salt outside so that they become less turgid. All right. Then we also talked about mesophyll cells full of chloroplasts. Those of you who did part C, you got to see this chloroplast in the elodea. You can see the cells expanding, contracting. It's really fun. Whether or not you're getting the credit for it, make sure you do it. A little announcement about labs, by the way. If you have a design due yesterday, and you send it to me at 12 o'clock, you're not going to get comment and time to fix it. Are you with me? Then you come to class, design is not approved, you waste a day. Are you with me? Design is due by 12 o'clock, but if you send it at 12 o'clock, you're not going to get feedback in time to change it if it's fucked up. Are you with me? Keep repeating that to you guys. Deadlines are not the day to turn in things by. They're the last day by which you can possibly turn it in. If you do it that way, you'll get feedback, and that's dangerous. Because that's all the last chance you got to submit it, right? One design is necessary per group in order to be able to do it, right? But if today you all do it as a group because you have a design that's been approved, that's great. But then the other people who did not submit designs don't get a grade unless their design is submitted, handwritten, is submitted by tonight. So that's the way it works. Prelab for each part. Data collection for each part and design of each part is due the day of you do that part. Then the report's final report is due 1 week after it was in the calendar that you have to do that part. That means you can do it late up to a week. So for example, Friday, right? It was when the reports for part C and D were due. So until Friday you could do it. Of course, if you did it part C on Friday, you have to finish up a report really quick to submit it by Friday. There's a folder that says lab report writing. Then it has instructions on how to write lab reports. And there's also a video lecture series in there where I talked about how to write lab reports for those of you who don't know how to do it. you're going to get feedback on having written or not written a good lab report as I start grading the ones we have submitted for Lab 4. It started last night, so you're going to get feedback on that. But then you're also going to peer review each other. Those of you who submit reports will get someone's report to evaluate. And in that process will help you learn how reports should be done. Because when you're trying to grade someone, you have to follow the rubric. And that way you say, oh, okay, that's the rubric I'm supposed to follow. That's the whole point, to do that. Each lab that you do, each experiment that you do has to have that. As far as, almost forgot to make these announcements. As far as the general pre-lab, all right? That's before the very first part, and that's where all those questions are due. For Lab 13, it says in the instructions that you can turn in those questions at any point during the lab, even by this final deadline, and that's fine. Just make sure that when you share with me, you say, anytime you share something with me and you're updating the lab document, you should always say what you're adding to it, because that's what I'll grade when I click on the link for it. All right? So where we left off last time is talking about the pigmentation. So you see that chain that I was talking about. So one pigment receives the sunlight, dumps electrons to the next one. That one gets hit by sunlight, dumps the electron to the next one. Every time the electron gets hit by the sunlight, it gets excited and it leaves the pigment. And it goes either to an electron acceptor or to the next pigment. And in that chain of pigmentation, eventually water has to come in to replace the electron that the first one in the chain or the last one in the chain is losing. All right, so that's water has to be split. But notice that the majority of these pigments is designed to make sure that most of the light is absorbed. Why do plants focus on blue light? Anybody has any idea why? Yes, Enzo? It's the most energy carrying one. Low wavelength carries more energy. But then why carry the orange and the red? Notice that the yellow and the green are barely carrying. That's why most leaves are green, because it's what reflects back from the heading of the leaf, right? But why is it they're carrying the red if it's the lowest intensity one? Think about evolution, right? The other reason also is that why not catch everything? If that's more energy, the better. You thought about that? Why did evolution select against the carrying any pigmentation that would carry the green and the yellow, or carry every light at the same level? What would happen if you were capturing all the light? What color would the leaf be? Nope. Black. What's the problem of being black? You overheat. What's the problem of overheating? Just water. Your enzymes don't work very well. And there's also photorespiration, which hopefully we'll get to talk about today. All right? Now, okay, so that we understand why not capture everything. So we understand also why capture the blue. But why capture the orange and the red? So what time does photosynthesis happen? Light reaction. When does it happen? During the what? Okay, during the day. Is the sun out? Does the atmosphere always have all the colors of the sun? Or is that part of the day where all you have is orange and then in the sunlight? You see? This allows plants to keep working late and early in the day. Are you with me? So any plant that evolved to capture that evolved and became advantageous, right? Also notice that in some places where seasons happen, leaves change color. It's almost like they lose, they stop absorbing that, right? And so they start giving out the red and the orange to you, right? So the production of certain pigments stops on those at those plants when it makes no sense to long collect it because of changes in the atmosphere. All right. So, anyways, let's actually talk about the light reaction. Okay. So then the light reaction, and again, remember I have a little animation of this, and so hopefully after I'm done explaining, I'm gonna show you the little animation. But basically, it's a chain, right, with two stations to bump electrons to higher levels of energy. So remember, when you see these stations, it's really a group of pigments. But the primary pigment of photosystem 1 or in photosystem 2 are, there's this one is P680 system, and it's actually, I don't want to teach you the wrong thing. Yeah, the first one is P680, the second one is P700. That just means the wavelength of light that's mostly absorbed there, all right? And one of them has more chlorophyll A, and the one who has chlorophyll A and B are in more presence. But basically the way it works, and I like this one best because it shows you what is actually happening in terms of energy level. Light hits the photosystems, right? The pigments kind of bounce the energies between each other, and electrons just keep jumping from pigment to pigment. Eventually, it all gets dumped to the P680 chlorophyll right in the middle, and those electrons get bumped to a higher level of energy. Now, when I have time, I usually do a little play with this so we can all learn it. But if we have time at some point in the year, I'll walk you guys through it. But then, basically the idea is this. Finally, the electron leaves the photosystem altogether, and it goes to an electron acceptor, which then dumps into another electron acceptor, which dumps into another electron acceptor, which dumps into another electron acceptor, and so forth. But in between, as it goes to plasmaprenome, to the cytochrome complex, and then to this one, in between what's happening is that the electron is being picked up, and a hydrogen is picked up and carried across. So as you go do this, a hydrogen is actually carried across. Now, in addition to the hydrogens which are carried across through this process, which we'll see in the video in a second, right? You also, by splitting of water, produce more hydrogens. So hydrogens are produced when water splits, right? And hydrogens are produced, also pumped into that space, right? Notice also that it's being pumped into the interior of the thylakoid, right? So it's inside the thylakoid that water gets split, and it's into the thylakoid that the pumping is happening, right? So they're making it inside of the thylakoid full of hydrogen, low pH, up to 10,000 times less pH, so like 10 to the negative 4, right? So they got a pH of like 4 instead of a pH of 8, right? So it's like bigger difference. That creates a potential on the thylakoid for diffusion. So the pH, they want to get out of there, but they can't because membranes are selectively permeable. See, this permeability is so important. Charged particles can get through the membrane, and so they're like trapped in there, but they want to get out of there, right? Notice also that a second bump of energy after the pumping happens, because remember, the pumping happens in between here, right? Now, see how this says ATP produced to the motion force? We're going to get to that second. No ATP is actually produced during the pumping, right? That's just something else. We'll explain that in a second. What they're saying is that when you pump, you end up creating the gradient that eventually will power the ATP production. But in between, once the electrons hit the second photosystem, light is absorbed again, it bumps them up again. Except this time, as they degrade, they do not fall into another photosystem. They fall into NADPH, right? NADP, which becomes NADPH, and therefore charges the battery. So this process will create a proton gradient, which eventually is used to produce ATP, and it will charge at the same time the electron carrier. Does that make sense? So the goal of this, pay attention because when we do cell aspiration, you're going to see how it's the backwards of this, right? So the process bumps up electrons each time it gets to the photosystem. The first time to create a proton gradient, which gets used to make ATP, and the second time to charge up the battery, right? Now notice that if you don't do both things, you do not charge the battery. All right, now how does this ATP get produced? Because the gradient is there, and then there's a gate. That's the only place it can come through, this gate. It's a channel to allow them through. But in the channel, there's kind of like literally a turbine. So this molecule is the shape of a turbine. So as the protons pass through, they spin a turbine, and then ATP gets produced by capturing the kinetic energy of the rotation. It's literally a molecular little hydroelectric dam instead of water flowing through the turbine. It's protons passing through the turbine, right? Now, of course, then the protons go back to the stroma, only to be pumped back into that by the next set of light reactions. Does that make sense? Now, this is happening throughout the entire day. So the entire day, NAD is being made, ATP is being made, right? And the plants do an excessive amount of ATP and NAD because they want to continue doing the carbon cycle during the night. So they produce an excess of NADPH and ATP during the day so that even during the night, they can keep doing what they're doing. Does that make sense? All right. There is a second process that's cyclic. So it's similar to the other one, except it only uses the second photosystem, which is actually called photosystem 1, because we think it's the first one to evolve. So we think that this stuff did not exist at first. You see? So then it only uses the actual photosystem 1. So basically imagine taking this out and taking this out, but still using this photosystem to keep doing this. Right? So all of that can do is produce ATP. It does not produce the electron carrier at the end. And it does not use photosystem 2, which evolved later to add the layer to this whole thing. Here's the photosystem 1. what it happens. So again, the ping pong of electrons happens in the carriers. Eventually gets dumped to the chlorophyll, and then as the reaction center sends that to the actual carriers, all it does is it powers that pump of protons. So all you really do is create a proton gradient, which can then be used to make ATP. But with this process, all you really do is ATP, right? So that means, going back to this, what's actually happening in thylakoid every single second is that you're making this. So you're doing this, and you're doing this all the time. Now the biggest difference between cyclic and non-cyclic is that the electrons that get bumped out bump back in. So this also does not need to split water. So you see, you can do this in the absence of water. No oxygen produced either, right? That's why we think it's more ancient. This precedes the production of oxygen through in photosynthesis. Are you with me? And we also see a lot of types of ancient of bacteria that do these. So there's a lot of bacteria that don't produce oxygen, but do this process to produce ATP. So they have electron light capturing mechanisms to production of ATP without the second mechanism that ends up producing oxygen. Does that make sense? And so we know this is more ancient because it's a highly conserved core process among a certain group of bacteria. So, but the other process of having photosystem 2 involved is less conserved. So we know it came later. If this is more ancient, it would be present in more bacterial groups. Does that make sense? Also remember the thylakoid, the chloroplasts are technically little endosymbiotic organisms, right? So technically all the photosynthesis that happens is made by bacteria, right? Technically, because so all bacteria is really what does it. Now, with that cyclic power of production of ATP, and by the way, the actual ATP produced during cyclic is not really used for this for the cell for extra. No, the cell can't survive on that. It's too little ATP. All right, it's actually necessary because if you do the math, you can see that the number of ATP produced by the non-cyclic is insufficient to power the Calvin cycle. So you need that extra cyclic to actually do it as well. The Calvin cycle is this complicated cycle which goes the actual understanding of the steps, intermediates, and enzymes associated with it goes beyond the scope of what AP biology expects you to do. But you do need to understand the basics of it. There's an important molecule called Rubisco, which has a high degree of association with carbon dioxide. It likes to attach itself to carbon dioxide. So Rubisco attaches itself to carbon dioxide. Now, carbon dioxide is a one carbon molecule. This Rubisco, all right, so you can see here, 1, 2, 3, 4, 5 carbons. You see that? So we have a 5 carbon molecule connecting to 1 carbon, so you got 6 carbons. But that intermediate 6 carbon molecule is very unstable and almost immediately splits up into 2 3 carbon molecules. You see that there? Also notice the phosphate groups, which are over here. That's going to be important. All right? All right? So then what happens is that now the next step is that for each, now everything that happens from now on is times 2 because you have 2 of them. Kind of like glycolysis. After you split the pyruvate, everything happens 2 times. Are you with me? Now, what happens is that ATP gets absorbed to create, now this is called carbon fixation, the first stage, because that's what you're doing. You're capturing carbon dioxide. The second stage is called, basically you're doing reduction, because you're dumping electrons, which means you're reducing the molecule, right? So electrons from ATP and NADH, the batteries, will then be dumped into this molecule together with the hydrogens and phosphate groups to charge the molecule with more energy, right? In this series of reactions that happen between here and here, what you're doing is adding energy to this molecule. The final product of this is production of a molecule called G3P. All right? This molecule is what we need to make macromolecules in the cell. This can be used to make sugar, protein, amino acids, a bunch of different things. It is the actual core unit that can be used to produce any of those molecules through a series of other biochemical pathways. Now what's interesting is that, so remember we had 1 5 carbon molecule, right? Split into 2 3 carbon molecules, right? And eventually you're also going to have 3 carbon molecules over here. But this actually has to happen 3 times, all right? So the whole thing has to happen 3 times. So 3 of these will produce, therefore, 6 of those. Are you with me? For every 6 of those that are produced, you produce 6 of those. Are you with me? And then, with 6 of those, 1 of them goes out of the cycle. The other 5 just go back to regenerate the 5 of those that you need. Right? So basically, so you need 5 times this 5 carbon molecule. All right? And then you have to do this 3 times to make this work. So in the videos, if you're interested, I go into this in more detail. This next phase here is just a regeneration phase to produce the Rubisco again. All right? Now, to produce the Rubisco again, 5 of the G3Ps produced will be used up to reproduce those molecules at the expense of yet additional ATP. So that's what needs to happen. This process was discovered by a biochemist that spent 20 years of his life studying reaction by reaction, step by step, by deactivating enzymes until he figured it out. And then he got the process named after him and a Nobel Prize to go with. Right? But this is how all of our macromolecules are made, because it produces the G3P, which is the key for production of macromolecules. Okay, so the first stage, this is actually trying to summarize it for you. So you got 3 molecules of Rubisco, 3 molecules of carbon dioxide, which ends up making 6 molecules of that intermediate. Then you have the charging phases. You're going to dump 6 ATPs because it's 6 molecules, 6 NADs because it's 6 molecules, to produce 6 molecules of G3P. But out of those 6 molecules, 5 molecules go this way to recharge the 3 molecules of that thing, right? And meanwhile, 1 molecule actually gets produced, right? So you actually kind of have to spin this so 3 carbon dioxide molecules get captured to produce 1 G3P molecule with 3 carbons in it. Makes sense. But of course, glucose is 2 of those G3Ps put together, because glucose has 6 carbons. So you actually have to spin the cycle twice, right, to actually make 6, right? And therefore, 2 carbon cycles involving 3 molecules to start with are going to be necessary to produce 1 molecule of glucose. But remember, that's not what they actually produce. Now, too much heat, lack of water, the quality and color of the light, right? These are some factors that can affect the rate of photosynthesis. The temperature can affect it, the color quality can affect it, the amount of water can affect it, all right, and so forth. Therefore, if you look at places that are too hot, not too much photosynthesis. If you look at places which are hot and humid, lots of photosynthesis, rainforest, right? If you look at oceans, it's going to be near the top where all the sunlight is at and the nutrients are at, and you're going to have a lot of photosynthesis, right? So forth. Now, the problem is that with Rubisco, which is our friendly molecule that captures carbon dioxide, is that it also is like oxygen. So it has almost nearly equal affinity to oxygen, but that's kind of stupid because I want to capture carbon dioxide, not oxygen. So this idea of photorespiration is the idea of an accidental capture of oxygen instead of carbon dioxide by Rubisco. What that does is it kind of breaks the carbon cycle, and then the cell has to spend some energy to rebuild the molecules and restructure the carbon cycle that's happening in the stroma. So every time one Rubisco molecule associates itself with oxygen, right? So then you go down this way, and then you have to spend NAD and ATP to recharge Rubisco without actually getting G3P out of it. Are you with me? So the cells don't want to do photorespiration, because what you do is you do a different cycle that still consumes the energy without producing the energy molecule that you want it to. Right? The problem is, going back to the structure of the plant, is that in the chloroplasts, light reactions are happening right here, and the carbon cycle is happening right there. But the light reactions produce oxygen right next to the place that's capturing carbon dioxide. So that means it creates this problem of photorespiration, which is not a big problem unless there's too much light, which sounds good. Photosynthesis increases, right? More light. But too much light actually increases the rate of photorespiration, especially if there's less humidity. Are you with me? And it's very hot. So in very hot environments where light is abundant, but there's less water and more heat, it actually increases the rate of photorespiration. So then plants have developed adaptations to save themselves from it. Two primary adaptations, C4 and CAM plants. The C4 plants, what they do is that they look what they do. The mesophyll cell, remember, are these cells on the spongy parenchyma, right? So they are going to be doing the capturing of light and all that stuff, right? But then it is bundle sheath cells, these ones here, which actually do the Calvin cycle, right? You see that? And the actual capture of carbon dioxide is not done by Rubisco at first. It's done by another molecule. That eventually, that's actually what happens right here. It goes beyond the scope of what you need to learn for AP biology. But on the mesophyll cells, a different process captures carbon dioxide. That molecule that's actually capturing dioxide, this PEP molecule, doesn't have the association problem with oxygen, right? And what that means is that you don't actually, the cell that's producing the oxygen, because that's the one that's doing the live reaction, right? That has full of chloroplasts and stuff. That one is producing oxygen. That's also capturing carbon dioxide here, but with a different molecule that does not have the affinity to oxygen. Does that make sense? Eventually, that goes to this little cycle that eventually dumps into malate, and it's the malate that's carried across a gap in the cell wall, right? Which is very important. This gap exists between plant cells to allow that to happen. Okay, this gap, I forgot what it's called. I think it might be called Plasmodesmata. I'm not sure about it, right? But this gap will actually allow them to go through it, and it's the malate that dumps the carbon dioxide in the Calvin cycle, which does not happen in the same place that oxygen is being produced. So what the C4 actually achieved is geographical separation of where light reaction happens and then where Rubisco is. Are you with me? But that means that the molecule that carries the carbon, it's 4 carbons at a time, malate, that's why it's called C4. This adaptation, which is present in a lot of grasses that are grasslands of hot environments, allows these plants to survive more in these hot environments because they reduce the photorespiration by separating the molecule that captures carbon dioxide in the Calvin cycle, Rubisco, from the oxygen that gets produced in the mesophyll cells. But that requires the evolution of a separate biochemical pathway to capture carbon dioxide, only to transfer it to a separate cell. So that means that's an advancement, right? Meanwhile, the cam plants do not geographically separate. They still do the photosynthesis in the mesophyll cells completely, though both processes. However, they only do the Calvin cycle at night. And at that time, they close the stomata. Right? Sorry, they open the stomata, sorry. Correction. So they open the stomata. But at that time, there's less heat, less water loss, less exposure to sunlight, and therefore, automatically, less photorespiration. Are you with me? So these plants, like the pineapple, the cactus, they all stop. They close the stomata during the day, which prevents water loss. All they do is light reactions throughout the day. And then at night, when it gets colder, when it gets less water loss problems, less exposure to light, which would increase photorespiration, they open up the stomata and do the Calvin cycle. So that's this temporal separation of the biochemistry, right? These advancements require a regulation of the enzymes that do the Calvin cycle, so they're deactivated in the presence of light, right? And how they do that goes, again, beyond the scope of AP biology, but it's interesting that these advancements exist. Now, photosynthesis, therefore, is a 2-step process, right, that involves light reactions to charge batteries, which then get used by the Calvin cycle, all right? So I'll stop here today, but I will also show, before I hit photoaspiration, for cell aspiration in another opportunity, I just want to make sure we did this on the EK. So we talked about during photosynthesis, chlorophyll absorbs free energy from light, yes, boosting electrons to higher energy levels. We talked about photosynthesis 1 and 2 are embedded in the membranes of the chloroplasts in order to transfer electrons into these molecules. And I talked about as a series of oxidation reduction reactions between carriers that eventually carries the thing across the membrane. We also talked about the formation of proton gradient, which is used to power the production of ATP through this chemiosmosis. And we also talked about the production of electron carriers, which are then used to power the Calvin cycle. Right? Someone's desperate to talk to me. We also talked about the fact that photosynthesis originally evolved in prokaryotic organisms, and the scientific supports that it's actually bacterial organisms that produce all the photosynthesis, and there's some that even do only the cyclic, right, which is more conserved than the non-cyclic one. Then the next thing is to talk about this, the rest of the cell respiration, right, which we'll have to do the next time we have a chance. All right. Now, before we move on completely to the lab with the last 5 minutes, I want to show you a little animation of this, that kind of, and then talk about it as it's playing. It kind of lets you. But this is why I said this folder is very important, right? Because we need a. It's very important to see biology in motion sometimes, and I think this is one of the examples of where it's extremely important. So let's see if we can do that. Are we in photosynthesis? Yes.
Was the PowerPoint on Google Drive, that one that you were just using?
This is my video lecture. The PowerPoint from that video lecture. And it's in the presentation. Now, the absorption of life during photosynthesis. So let's look at that. All right, so for the sake of my computer speed, I'm actually stopping the podcast. So make sure you watch videos from, if you're not here, if you want to review it. What I'm about to do next is show you animations from the videos in my tutorials folder.
Absorbs all the
All right?
visible light that strikes it. If it absorbs